We're learning a bunch of crap. This is the characteristics or properties of epithelial tissue. There's 4 types of tissues in the body: epithelial, connective, muscle, and nervous. As we learn the 4 types, the best way to learn them is describe them by comparing them on their structures with the other types of tissues. We're learning crap, which is cellularity, regeneration, avascular, and polarity. This is only talking about epithelial tissue. So first, when we describe cellularity, and I look at this picture here, it's showing all these cells tightly packed together. Remember, the dark dot in the middle is the nuclei, so this is one cell here, and I look at that plasma or cell membrane, it tells me it's another cell. So these cells, relative to the other types of tissues, are closely together. So when we look at connective tissue, we'll see that the cells are more spread out, and there's material that the cells made in between. We don't see that in epithelial tissue because the structure creates a function. We're trying to provide that barrier, because remember, our skin is epithelial tissue, and then also to control permeability. So cellularity, tightly packed cells relative to the other types of tissue. Regeneration, when we talk about regeneration, it just means out of the other types of tissue, epithelial tissue regenerates or goes through mitosis the most. When we look at our layers of the epidermis, there's 4 to 5 layers, but that changes over the course of a month. It sheds off very quickly. When I burn my tongue, you feel that sensation for a day or two, but then it goes away, right? Because it repairs itself so quickly. Versus my brain, versus my heart, those are different types of tissue. They don't regenerate, sometimes not at all, or they regenerate very slowly. Avascular, A meaning without, vascular meaning without vessels. So they don't have direct blood supply. When I look at the picture here, the skin, this is the epidermis. Below it is connective tissue, and this is the dermis. When I look at the dermis, I can see those blood vessels. This is the blood supply for that epithelial tissue on the top. So I don't have direct blood supply in the epithelial tissue. It's supported by the underlying connective tissue. And then lastly, polarity. Polarity is usually the most difficult for students to describe, but it's 2 distinct ends. When I look at other types of tissue, you can't always see like there's one end and here's the other end. For connective tissue, we clearly see that. The best example is when we look at the epidermis, that I have a top layer and a bottom layer, just like a battery has a positive and a negative end. And we call that the basal surface and the apical surface. The apical surface is that free surface or free border where it's exposed to the environment. The top of the epidermis or superficial layer, this is the apical surface. The deep portion of it, where I have the base of the cells, this is the basal surface or basal border. We'll see what's called a basement membrane or basal lamina. So the end of the tissue as I go deep, that's my basal surface. Whereas when we look at other types of tissue, we don't see a distinct top and bottom. But when I look at a tube, it's the same idea. Remember, some of our glands and our tubes are made up of epithelial tissue. If I look at a tube, let's say this is of the kidney, and we're looking inside of the pipe, where the fluid is running, and we call that hole the lumen, but where I touch the inside of the cells, this is the apical surface. And remember, I said it's exposed to the external environment. So if I drink a liquid, say coffee, it still goes down into my body, reaches the urinary system, and is running through these tubes. This is still what we call the free surface or apical border, apical surface. So that edge, and then the base, like the outside of the tube or outside of the hose, this is the basal surface. So we can describe it as like a top layer, a top and bottom, like the skin, or the inside and outside of a tube. So 4 characteristics of epithelial tissue: cellularity, regeneration, avascular, and polarity. This crap is unique to the other 3 types of tissue. Functions of epithelial tissue. There's 4 types of tissues in the body: epithelial, connective, muscle, and nervous. In a previous video, I talked about the characteristics or properties of epithelial tissue because it helps us start to explain its structure that leads us to its functions. So this is just a quick outline of functions. We can use the skin, specifically the epidermis, as a way to describe these functions. So quickly just looking at what does epithelial tissue do? Well, I've talked about it that it's the windshield of our body, so it does provide that physical protection. When we look at our skin, it's not only a physical barrier, but then it also helps us to protect from things like microorganisms, right? It's that windshield, that barrier. So physical protection. How do we do that? The cells are tightly packed together. That's our cellularity, which leads us into control permeability. Remember, 2/3 of our body is water, so we have to control how much water is leaving our body. We see it leaving through perspiration, but we also have to control what's going in and out. And I see that, let's say I throw a can of paint on you. It doesn't go right into your skin, depending on the chemical makeup. It takes some time to get through unless I wash it off. And then I have the ability to wash a lot of things off, to decontaminate myself. So we control that permeability. And then I see that when I'm just in water, say I'm in a hot tub or jacuzzi, that my skin will start to wrinkle, right? That's our stratum corneum, that superficial layer that's doing that, but it's trying to control the permeability. We'll learn about different ways that we have sensation. There's specific cells that are specialized within the epidermis called Merkel cells that help us with just light touch to be able to understand, like on our fingertips. If I think about my lips when I make contact with things, and then we'll learn about collections of cells called corpuscles that help with light touch and with deep touch. Specialized secretions. We've talked about the modes of secretions for exocrine glands. And when we look at the skin, I see that I have these little flowery structures around the hair. These are called sebaceous glands. They specialize in what they secrete. It's the sebum or oil of my skin. And then I have eccrine or American sweat glands built in that provide that watery perspiration to come up to the top to cool my skin. So I have specialized ways to secrete a product. And then filtration, excretion, absorption. I kind of move on and just show the kidneys, because when we learn the urinary system, we'll talk about the functional unit of the nephron. And when we look at the nephron, it has the ability to, with its tubules, to filter out typically excess salts and waters. But when we look at the filtration process, it has the ability to then to essentially like a coffee filter to filter out what I don't need. And then I excrete that waste, or then I could reabsorb it. A lot of times when we talk about absorption, when we look at our lungs, it's made up of simple squamous epithelium at the alveoli. Its ability to absorb is based on its functionality and structure of the tissue. So basic functions of epithelial tissue, we're comparing them again to our other 3 types of tissue, but understanding the basics of the functions leads into understanding how its makeup exists. Classifications or types of epithelial tissue. Previously, we talked about the functions of epithelial tissue. We talked about the characteristics of epithelial tissue. Now we're describing really the 2 ways in how we name them, but it's based off their structure. We name them 2 ways. First off, the layers of the cells. When we covered our characteristics, one of the things we talked about was polarity, that it had 2 distinct sides, a basal surface and an apical surface. How we describe that is the number of layers. So you see across the board here, this says all simple, and this says stratified. Simple means it's 1 layer of cells. Stratified means it's 2+ layers of cells. And what it means is what it's sitting on that basal surface. So when I look at this layer, underneath here is the basal surface, and then there's 1 layer of cells. 1 layer of cells, but when I go down here, this is 2+ layers of cells. And it's all based on the structure creates the function. Simple, 1 layer of cells makes it easier to move things across. Osmosis, diffusion, filtration. When I have stratified, typically it's more for protection. When we go back to our layers of the skin and we learn the epidermis, that's stratified epithelial tissue because I need that protection because it's the windshield of my body. Then we describe it based on the shape of the cells. So squamous or squamous, cuboidal, and columnar. As we look at the shapes of the cells, squamous is referring to like scales. So when you think about the scales that come off of a fish, thin, flat cells are called squamous cells. So they're thin and flat for a reason. Thin and flat allows for the movement again of materials. When we look at the alveoli of the lungs, to be able to breathe in, I want to make sure that oxygen carbon dioxide exchange is very efficient. So I use squamous cells for that. As they get larger in size, now they become cuboidal. So cuboidal cells, look like a cube or a square, and we get a little bit more room in that cell to allow for more activity. So typically we see cuboidal cells now are more active, and where we see them as areas of secretion. So they're secreting a product. So when we look at some of our sweat glands, they'll have cuboidal cells. When we look at the thyroid gland, it can have cuboidal cells. They're secreting something. Versus squamous is allowing just for movement. When we get into columnar, when we look at a microscope slide, to me they tend to look like fingers because they're just longer like rectangles. So all of these are individual columnar cells. More room in the cell, which means more activity. We tend to see them where you have more complex chemical reactions like my digestive tract. Stomach, small intestine, large intestine is all simple columnar that lines those linings of the tubes because we're going through the process of metabolism and breaking down food. So we have columnar. Transitional means it can transition back and forth to have multiple types. It can be squamous, cuboidal, sometimes columnar, but a mix of the three shapes. What that allows me to do is movement. So when I have different sizes, it allows for things like stretching. So we typically use the example of the urinary bladder. As it fills up with urine, it's expanding. And the different types of cellular shapes allows for movement. So transitional is a mix of the three. So then we go stratified squamous, stratified cuboidal, stratified columnar, just 2+ layers of the cells. And then lastly, we'll describe pseudo-stratified and pseudo-false stratified, so they look like they're stratified. To me, the easiest way to think about this is like when you're looking downtown at buildings, they're all different sizes. So it looks like there's multiple layers, but they're all connected to the ground. And in this case, it's connected to that basal surface. So they may look like different sizes, but they are all connected to one side, the basal surface. So that's pseudostratified. The other thing we'll see is they have cilia. Cilia are these little spiky things running across the cell surface. Cilia allow for movement. When we look at microvilli, that's for absorption. Cilia allows for movement. Columnar cells, because they're more like columns. So pseudostratified ciliated columnar epithelium, we tend to call that PSCC. Best example of that is we call this the respiratory epithelium. We've talked about different types of cells and tissue membranes. We talk about mucus that's produced by goblet cells. We have this in my trachea, the ability to bring up the stuff that I breathe so it doesn't harm my respiratory system, my lungs, and I can spit it out or I can swallow it. Pseudostratified ciliated columnar epithelium is where I see this in the respiratory system primarily. So we have them described based on their number of layers, simple and stratified, and then the shape of their cells. The structure creates the function. Remember, this is unique specifically to epithelial tissue. Later, we'll talk about the makeup of connective, muscle, and nervous. This is simple squamous epithelium. So far in previous videos, we talked about the characteristics of epithelial tissue. We talked about the functions of epithelial tissue. We talked about how we name them based on the number of layers plus their shapes. And now that finally leads into we can study the individual types. So we're starting with simple squamous epithelium. Remember, simple meaning one layer of cells. Squamous meaning thin and flat. So use the three terms to describe the tissue. Where do we see simple squamous epithelium? Remember, simple is to move things. Squamous is thin, flat cells. Good example is alveoli of the lungs. So I exchange those gases. When we look at histology or the slides of the microscope for simple squamous epithelium, I use this little picture to go back to show the arrow. We are looking at it like you're looking at the tiles of the floor. So other microscope slides, when we look at epithelial tissue, would look more like a side view. So when we look at the microscope, it looks like it's multiple layers, but not when you look down on the floor, just looking at the top of the tile. So you're looking at essentially the apical surface. 3 different views depending on what microscope slides you have. It's just like anything else. I look different than you, but I still have a nose. It just looks different. So these 3 slides are the same. It's just all simple squamous epithelium. So we're looking at the top of it. All of these are individual cells. So I have, I don't know, 100 slides or 100 cells in this picture here on the top left. Remember, the dark dot in the middle indicates the nuclei, and then the surrounding line around it is my cell membrane, plasma membrane, or plasma lemma. Any of those three terms work. And then we're just looking at the material inside the cell, which then is the cytoplasm. If I go down to this one, cells a little spread out in this microscope, but remember we have cellularity, tightly packed cells. So all the individual cells, and I can see them distinctly because I have that dark dot in the middle, the nucleus, the cytoplasm within it, and then the boundary, my cell membrane. I like this one the best. I think it's the most consistent slide you'll see across text, but this is all showing that cellularity. And here's some nice cells here as I wrap around the cell membrane, the middle dark dot, the nucleus, and then cytoplasm is the material in it. If we had a more powerful microscope like an electron microscope, we could see some of the organelles potentially. But for a light microscope, it's going to be pretty universal that we're just going to be able to see the nucleus and the cell membrane so we can see from cell to cell. Simple squamous epithelium. Simple cuboidal epithelium. We've looked at simple squamous epithelium. Now we're looking at cuboidal. Remember, simple, one layer. Cuboidal, they tend to look more like cubes or squares. To me, I remember they look more like pearl necklaces. When I look at simple squamous, those cells, in my mind, I see a bunch of fried eggs. So as I look into microscope slides, I try to change it in the sense of like, what do I actually see to help remember what I'm looking at inside the microscope? To me, these look like a bunch of pearl necklaces, just at different magnifications. Where do we see simple cuboidal? Remember, areas of secretion. We see them in ducts, in glands. We see them things like thyroid secreting something. So at a thyroid, secreting hormones. So areas of secretion, so more active cells. So when I look at this part of it, we're looking at a follicle hill, but all these cells that wrap around here, individual cells, and I can see that based on the nuclei, are simple cuboidal epithelium. If I expand on this, makes it a little bit easier. Remember, to me, I said they kind of look like pearl necklaces, but I can see those nice round nuclei. Those indicate cell to cell. And this is looking at inside of a tube. So this is inside of the tube, inside of the tube, inside of the tube. Each individual cell is designated by that nuclei. The nuclei tend to sit in the middle of cuboidal cells, and it's taking up most of the cell in this case. Remember, the outside wrapping is the cell membrane, or plasma membrane, or plasma lemma, and then the cytoplasm is the material within it. If I expand on this one's nice. When I say a pearl necklace, to me this looks like a pearl necklace. And each individual cell is a cuboidal cell. Remember, we learned our two sides for polarity. The inside area, that lining is my apical surface. The outside of the tube is the basal surface. Each individual cell has the nuclei in the middle, so those dark purple structures, nuclei. The boundary, can't see well for all of them, but you can see that line there, and that indicates the cell membrane or plasma lemma or plasma membrane, and then the material inside, the cytoplasm. So simple cuboidal epithelium. Simple columnar epithelium. We've looked at simple squamous, remember, thin and flat cells. To me, they look like fried eggs. Simple cuboidal, areas of secretions. They look like cubes to me. When we put them together as a tube, they look like a pearl necklace. Now we look at simple columnar. They tend to look more like rectangles, but to me, a lot of times I see they look like fingers or stubby fingers. Three different views of it. So we look at a pictogram of it. One thing I just want to share with you is when we look at more of columnar, the nuclei tend to be closer to the basal surface. Remember, we learned our polarity. There's two sides, apical surface and basal surface. And basal surface is that deeper foundational layer. And I look at those nuclei and we see them closer to the basal surface. So if I take that picture and expand it down here, I'm going to draw the edge. This is the basal surface here. So going up, this is all my cuboidal cells leading to the apical surface. And you can see all those nuclei that are hugging close to the basement membrane. So to me, I see more like stubby fingers, not squares or pearl necklaces. And then you have a lot more cytoplasm. So you have a lot more room in the cell. Why? The structure creates the function. I have more complex chemical reactions. Where do we see this the most? We see it in our digestive system, the gastrointestinal tract, stomach, small intestine, large intestine. So this view shows it like the lining here. So the food would run along here. If I go to this picture on the right, what this is showing you is the layers of the small intestine. So we're looking at epithelial tissue, and then there's connective tissue, and there's muscle tissue. We'll come back to that when we get to histology of the small intestine. Right now we're just focusing on this ribbon here on the top. And when we see hills and valleys, this gives me surface area. It's one of the key things we'll talk about in the course. The more I have hills and valleys, the more I can put things through. So this is microscopic for that food to drop down in here and start to absorb those nutrients. So when I look at this lining and I just take one of these fingers here, just this one, that's looking at this here. So go back again. This lining is just this lining. So it's wrapping like this, and that's where I can see all these columnar cells individually. And how do I know which ones they are? Because I can see those nuclei. In the middle here, almost like you're looking at a finger, the meat of the finger is connective tissue, whereas the lining of the finger, that's my simple columnar epithelium. So those individual cells are going like this, and so I have a lot of cytoplasm here. I can't see too well the plasma membrane, but I know cell to cell just because of those individual nuclei. So it's wrapping around. When I go to this picture, it looks inside of a tube. Looks pretty close to looking like cuboidal cells, but they are longer, more like stubby fingers. So this is all individual columnar cells, and notice those nuclei are closer to my basal surface. So my basement membrane is that structure, that surface, basal surface. Inside of the tube or lumen, this surface is the apical surface. And I like this picture because it clearly shows nice round nuclei, and then those lining of the cell or boundary of the cell, my basement membrane, my plasma membrane, or cell membrane. So this is simple columnar epithelium. Stratified squamous epithelium. So far we've looked at simple layers, simple squamous. Remember I said they look like fried eggs to me. Simple cuboidal, squares or cubes. They look like pearl necklaces when create a tube. Simple columnar, my fingers. And now we're on to stratified squamous epithelium. Remember, stratified means 2+ layers. As we start to learn the integumentary system first, we cover stratified squamous epithelium when we're talking about the epidermis. So this top layer or superficial layer of the skin, the epidermis, that's the layer. The type of tissue is stratified squamous epithelium, one of our types of epithelial tissue. To me, it always just like a nice pink purple ribbon. So if I expand on this, just this purple ribbon here is my stratified squamous epithelium. Deep to that, this is all getting into the dermis, which is the layer deep, and this is all two types of connective tissue that we'll cover later once we start connective tissue. So that ribbon, if I expand on it here, this is magnifying it from here up. This is my superficial layer of the skin where I can touch at stratum corneum. And that's where you can see that it's literally stacking of cells. And I just go by the appearance and know my order. As this dark purple ends here, this is my basal surface, which means as I go up, this is my apical surface. So stratified squamous epithelium, that nice purple ribbon. If I magnify it more, this is the end at that basal surface. Deep to that is my underlying connective tissue, but this is all stratified squamous epithelium. Just a nice purple ribbon. So just learning the terminology, the layer, epidermis, type of tissue, stratified squamous epithelium. Why is it stratified? For that protection. Pseudostratified, ciliated, columnar epithelium. That's a lot. We abbreviate it as PSCC. On your exams, don't abbreviate anything. Pseudostratified, ciliated, columnar epithelium. Remember when we looked at simple 1 layer, stratified 2 plus, pseudostratified. Remember, it looks like it is multiple layers, but all my cells are essentially connected to my basement membrane at that basal surface. It just appears that way. So there are active cells. Remember when we have those columnar cells, more ability to conduct activity. We often refer to PSCC as our respiratory epithelium, and I'm showing it because we see it in the trachea. And so I expand over here, this from here to the superficial side, this is all pseudostratified, ciliated columnar epithelium. All those nuclei are kind of sporadic to make it look like it's stratified. When we come back to this, we'll learn layers of the respiratory epithelium to look at this as actually cartilage, and then this is a connective tissue layer, the submucosa. Not learning that now. Just from here up, this is all PSCC. How can I see it? What's unique to it for identifying it is the cilia. So on the surface, this is where I am secreting mucus in my trachea, and this cilia is an escalator. It's moving my spit or mucus up so I can spit it out when I trap in particles that could impact my lungs. So as soon as I see that cilia, I know I'm looking at pseudostratified, ciliated columnar epithelium. So the air is passing in this white area here as I breathe in, and I'm grabbing onto the junk that I breathe. How do I do that? By way of goblet cells that produce mucus. Okay. As I expand over here, I'm using this picture to describe what we're looking at, but you can see all these round dots are my nuclei, and it's spread out to where it looks like now they're stratified. So from here up, this is all PSCC. This is my basal surface. This is the underlying connective tissue. But as I come up, now I can see all those little hairs that are cilia. And cilia's purpose is to move them across the cell surface. This is pseudostratified ciliated columnar epithelium. As soon as I see those cilia, I know I'm looking at PSCC.